Good morning, everyone. Today we'll be discussing about gene library. What is a gene library? A gene library is collection of different DNA sequences from an organism, each of which has been cloned into a vector for ease of purification, storage, and analysis. The gene library can be of two types. It can be either genomic library or cDNA library. So basically, what is the basic difference in these two kind of gene library? In case of genomic library, what we are cloning is the genomic DNA. Whereas in case of cDNA library, what we are using as a template or what we are cloning or what we are going to study will be the cDNA, that is the complementary DNA, which is produced by mRNA. Now, for the construction of DNA library, the size of the gene is very important because depending upon the size of the gene will depend your vector which you're going to use. Say, for example, the size of the gene is very large, then you have to go for a high carry capacity vector. If the size of the gene is okay, it's not very large, 500,000, then you base pair, then you go for plasmids and phosphates. We need to, as I talked about, that we need to be very careful about the size of the gene. So we have to be considering the capacity of vector also. We need to go for different kind of molecular biology tools like restriction enzyme, modifying enzyme, then ligases. All these will be required for construction of a library. And what kind of vectors we are going to use, that will depend upon the size of the insert and the capacity of the vector, and also what eventually we are going to do with this library. We are going to store it, how we are going to screen it. That all will depend upon the kind of vector which are being used to construct the DNA libraries. Now, what is a genomic library? If we talk about, then genomic library is nothing but it is the representation of the entire genome of an organism in form of small DNA fragments, and this is created by using molecular cloning. The genome size we know is expressed in terms of number of base pairs, that is, the DNA is comprised of three bases, that is adenine, guanine, cytosine and thymine, and they have different how much number of these bases are present, that makes the genome size. The size of genome is different in species, it is variable. Generally in complex system it is high, whereas for prokaryotic systems the size of genome is small. But yes, here we have to keep in mind the cDNA paradox. In prokaryotes, the structural genes don't carry any non-coding region which is intron. They only carry the coding region, whereas in eukaryotes, we have both introns and exons, the non-coding as well as the coding region. So therefore, the construction of gene library for eukaryote will be more complex because there is a lot of non-coding regions also which we will have to take care of when we are making a library or when we are going to get it expressed or checked or selected. The steps which are involved in construction of genomic library are isolation of DNA from cell. Here, the DNA is isolated from the organism or the cells of an organism for which the DNA library has to be constructed. It is digested into small fragments using the physical method, sharing methods, or we can use the molecular scissors like restriction enzymes. Then they are introduced into suitable vector and we get hold of the recombinant DNA molecule, which is then inserted into the bacterial cell for its further culturing and for isolating more amount of DNA from these bacterial cell. And for this we need a lot of transformation techniques to insert this clone recombinant gene into the bacteria. The DNA is isolated after culturing of these bacterial cell, and the collection of genomic library is what we get in the end. So here, these are the steps which are discussed. We are isolating the DNA and how it is done for prokaryotes and eukaryotes. As I already said, the DNA can be, we can break the DNA fragments. We can make the DNA into fragments by using physical sharing method or restriction enzyme digestion. Physical sharing includes pipetting and mixing, but generally it is not a very sorted method because it can lead to a lot of damage to the sample. So we go for restriction digestion. Here also we need to be very careful that we are having a partial digestion as well as complete digestion samples with us, so that we get different varied length of DNA fragments. This is a representation of construction of genomic DNA library. Here we can see that from the cells the DNA is isolated and bacterial vectors, that is plasmids, are taken up, and then both of them are digested with restriction enzymes and eventually ligated together and inserted into bacterial cells by transformation, and what we get is then collection of DNA library. That is the the basic clones we get in our hand. Selection of restriction enzyme. We know that restriction enzyme are of two types. They can be either overhang producing or sticky and producing restriction enzyme, or they can be blunt and creating restriction enzyme like SMA1. So that we have to keep in mind. Now, whether is enzyme inhibited by DNA modification, also the time of digestion, and what is the ratio of restriction enzyme which is required for the DNA. All these points have to be considered. So here the next step is introduction into suitable vector, wherein each fragment is different and they have unique DNA sequences. So they are inserted into suitable vectors, which can be either plasmids or bacteriophage vectors, and these vectors are digested with same restriction enzyme and sealed to, say, human DNA, for which we are going to do we are making the DNA, the genomic library using DNA ligase, and the resulting molecules are recombinant because now it is carrying the component of a plasmid as well as the gene of interest. This recombinant molecule is then inserted into bacteria, say for example E. coli, or you can also go for bacteriophage, that is a lambda genomic libraries if the size is very high. But here we are explaining the example of bacterial clones. The microbes are grown in culture. They are made to take up DNA. That is, if the DNA is inserted into them using transformation techniques, which can be either electroporation or with microprojectile, because they are high transformation efficiency. They give high transformation efficiency. So now we get the collection of clones which contain all the sequences which are found in an original source, including the sequence of interest. Now these all constitute what? They constitute the genomic library. Multiplication and production of clone is carried out by culturing these clones and then isolating DNA from them. Screening of clones is done by different ways. It can be done by expression screening, wherein you're looking for the expression of a particular product. It can be done by insertion selection and activation method, in which, say for example, a particular antibody gene has been disrupted due to this insert, so it will not produce, will have any more resistance for that antibody. Or it can give blue white selection method can be used, because it will give blue color or it will not produce a blue color depending upon the insertion inactivation. And one of the most sorted method for genomic library screening is colony hybridization. So why do we actually construct genomic library? So why we do it is that researchers can explore the genome of organism to learn more about its genomic structure and function. They can map the genome, identify the location of specific gene. This helps them to develop tests which can be used to locate genetic variation, including mutation. Also, it is useful in rDNA technology because we are able to genetically modify the organism. Also, it is the first step of any DNA sequencing project, and genomic library help in the identification of novel pharmaceutically important genes, which has a lot of significance in healthcare sector. So this is the shotgun sequencing method wherein genomic DNA from all the clones is isolated, and then it is fragmented by DNA, and we study the sequence of uh, the DNA fragments using computer, and here we can see that we are getting the sequence of all of the different kind of fragments, and then they are aligned together, and once that is done we get the whole genome. Coming on to cDNA library. For cDNA library, mRNA is isolated, it is purified, and it is checked for its integrity. Then it is fractionated, and enriched mRNA is then used to synthesize cDNA, and the cDNA ends are then treated. There is no CNA library made from prokaryotic mRNA because prokaryotic mRNA is very unstable. And of course, genomic library of prokaryotic organism is much easier to make because it contains all the genome sequence for the reason that they don't carry any intron. cDNA library are very useful for eukaryotic gene analysis. Condensed protein encoded gene library have much less chunk sequences. cDNAs don't have any introns, and it is very useful to identify new genes, especially the tissue specific or cell specific ones. Now, mRNA is isolated, and we know that mRNA of eukaryotes at the 5' have a cap, and at the 3' they have at the 3' end they have poly A tail. Now, this particular structure of mRNA is used to isolate it from among, I mean, we can get hold of mRNA in place of rRNA by using the matrix, which is oligo T rich matrix, and here only the mixture of cytoplasmic RNA is put loaded, and only that mRNA which has poly T tail, which is the mRNA, sorry, only that RNA which has the poly A tail, which is the mRNA, get attached to this oligo T matrix, and rest all get is washed off, and eventually it is this mRNA is eluted from this oligo T matrix, and now we have mRNA in our hand to make the complementary DNA. Now, to make complementary mRNA, there are traditionally different methods available. These are the three methods: the RNase method, the self-priming method, and homopolymer tailing method. We'll be taking up each. In case of rRNA method, what we do is, the principle of this method is that the complementary cDNA is synthesized using the reverse transcriptase enzyme to make a RNA DNA duplex. Now, this DNA strand is then nicked and it is replaced by DNA. So what exactly we are doing is that in this method, the first step is to anneal a chemically synthesized oligo T primer to the third poly A tail of RNA. The primer is typically 10 to 15 residue long, and it primes the synthesis of first DNA strand in presence of reverse transcriptase and oligonucleotides. This leaves a RNA DNA duplex. The next step is to replace the RNA strand with DNA strand. This is done by RNase H enzyme, which removes the RNA from this RNA DNA duplex. The DNA strand thus left behind is then considered as the template, and the second DNA strand is synthesized by action of DNA polymerase II. Coming on to the second method, which is the self-priming method. Now this involves the use of oligo T primer annealing at the polyadenylate tail of mRNA to prime first DNA strand synthesized against the mRNA. This cDNA thus formed has the tendency to fold back on its own, forming a hairpin loop. This results in the self-priming of the second strand, because from here only the second strand start getting constructed. After the synthesis of second DNA strand, this loop must be cleaved with a single strand specific nucleases, which we know are S1 nucleases, and this helps to allow the insertion of cDNA into the cloning vector. One of the serious disadvantage of this method is that this S1 nuclease can also cut extra sequence from 5' end. So that is very important to keep in mind. The third method is homopolymer tailing, and this approach uses the enzyme terminal transferases, which basically do polymerize the nucleotide onto the third hydroxy of both DNA and RNA molecule. What we do is that we carry out the synthesis of DNA strand essentially as before to produce a duplex of RNA DNA. We then use terminal transferases and a single deoxyribonucleotide to add poly tail to the nucleotides of the third prime end of both RNA and DNA strand. Now this results in a DNA strand basically which has a known sequence at the third prime end, which can be either poly C-TP or poly DATP. Poly C or poly A can be used. A complementary oligomer can be synthesized, which will help basically to prime the second DNA strand synthesis. This oligomer may additionally incorporate a restriction site also if we require, which can be used and can be helpful in cloning this double-stranded cDNA. So blunt end ligation of large fragment is not efficient, so we have to use special acid linkers to create sticky end for cloning. So in this process, what we do is that a strand special nuclease is required, and we fill the missing third prime nucleotide using Kleinow fragment of DNA polymerase and the DNTPs. We ligate the blunt end and linkers, and restriction and enzyme digestion is then carried out. Any vector with an E. coli R1, EcoR1 site would be suitable for cloning this cDNA because it has EcoR1 site at its ends. Now going for screening procedures, screening can be done by colony and plaque hybridization. It can be done by expression screening. We can also do hybrid arrest and release, and we can go for chromosome walking. So screening is a process. What is screening? Screening is a process of identifying one particular clone containing the gene of interest among the very large number of other clones which are present in the genomic library. So what we use this nucleic acid probe to screen the library based on hybridization with the nucleic acid, or we can analyze the protein product. Any one way we can go for. Screening libraries. Now for searching the genes of interest in a DNA library, hybridization is used. That is hybridization to identify the interested DNA or its RNA product. So here basically radio-labeled probe which are complementary to the region of interest are used. Now what are probes? They are an oligonucleotide derived from the sequence of a protein product of the gene. A DNA fragment, they can be oligonucleotides and they form a related gene of another species. Then the blotting the DNA and RNA on the membrane, that is the transfer of our either DNA or RNA on a membrane, and then hybridize the label probe with the DNA membrane or RNA membrane, southern or northern hybridization. Coming to colony and plaque hybridization, here what we do is basically we will explain the bacterial one for now. Bacterial colonies must are taken up and they are lysed to release DNA on the membrane surface, and then hybridization is carried out. We remove the unhybridized probe and then we can visualize it either by using X-ray film or by streptavidin biotin system can be used for this, or we can also use antibody to basically visualize the results. Also, there is another way in which it can be done is if you have phage, that is when your cloning is done in bacteriophage, then you go for plaque hybridization. Now, what are the difference between colony hybridization and plaque hybridization? Colony hybridization is a method of screening bacterial colonies with desired gene, whereas plaque is basically the screening method to use, which is used for the recombinant phage. Colony hybridization was developed by Briston and Hogness, whereas plaque hybridization was first developed by Benton and Davis in 1977. In case of colony hybridization, what is involved is identification of plasmid or plasmid based library, whereas for plaque hybridization, the phage DNA libraries are involved. In colony hybridization, we can lift the entire set of clones together, whereas for the plaque hybridization, several lifts are required. For colony hybridization, there are more background signal. For plaque hybridization, there are very less background signals. Coming for expression screening, we can also identify protein product of an interested gene by going for either the protein activity of this product, or by Western blotting using the specific antibody. Thank you.